can be done. Okay, so now let's have a small video which would be helping us to understand the DNA replication. Okay, all of you. DNA is a molecule that is made up of two strands twisted around each other in a double helix manner. Each strand is made up of a sequence of 4 chemical bases represented by the letters A, C, G, T. Okay, this is what we know. The two strands are complementary. This means that wherever there is a T on one strand, there will be an A in the opposite strand. And wherever there is a C in one strand, there will be a G on the other strand. Each strand has 5' to 3' end. The two strands run in opposite directions. So you can see this determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done with the help of an enzyme that is called as helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. RNA bases are added up. This enzyme makes small piece of RNA that is called as a primer. This marks the starting point of the construction of a new strand of DNA. An enzyme next comes as DNA polymerase that binds to the RNA and will make up the further chain. DNA polymerase can add up the bases only in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous manner because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called as the Okazaki fragments. Each fragment is started with a new RNA primer, followed by DNA polymerase that adds a short row of bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again and again. Once the new strand, new DNA is being formed, the enzyme exonuclease removes all the RNA primers from both the strands. Another DNA polymerase enzyme then fills the gaps that are there within the DNA molecule. And then you have the final enzyme, DNA ligase, that seals up the fragments in both the strands to form a continuous double strand. DNA replication is thus described as semi-conservative because each DNA strand is made up of one old strand, conserved DNA strand, and a young new strand. Okay, so I hope this is clear to all of you. Now, for your reference further, I have given some diagrams. This is the diagram that's there in our text.